started in a moment, but I did want to say hello, and please feel free to drop in the chat where you're tuning in from. If you are a classroom, if you are, you and your family, if it's just you and, your 3 dogs, that would be great also. We just love to hear where folks are tuning in from and who you guys are, if you will. So we're going to get started in a minute. Oh, fantastic. Oh, UMass Amherst. Nice. And
I do it.
Fantastic. Awesome. So obviously we have our University of Alaska Fairbanks folks joining, which we love.
Right.
It's so great to have you guys all on board. And oh, we do have a question. So Judy, if you want to I see your hand raised. If you want to put your question in the Q&A, we can make sure we get to that. Oh, Natalie, your dad is on. So the family support is here. Welcome. I see New Jersey. I see North Carolina. And I see California. This is so awesome. So we're going to get started because we have a lot to cover today. So first, I just want to say welcome, everybody. As you can see, our attendees are muted and can't turn on the video. I will go over this in a second, but the connectivity can be a little touch and go on our research vessels. So to save some of the bandwidth, we have made sure that is going on, or that the videos are off. So my name is Sarah Ecker. I work for the Office of Polar Programs. I'm a communications specialist, and I'm really happy to have you guys joining us. As we go through this event, please feel free to use the Q&A function. That's where we will be pulling all of our questions when we get to that part of our presentation. As I noted, connectivity can be an issue sometimes on our research vessels. And so you might notice some freezing when we get into the presentation, but bear with us, our team will be back. They are actually in the eye of a storm. So if you see some swaying or you see some things moving around, that's what's going on. They are actually on a research ship. So the other thing I wanted to say again, if you're just joining us, please let us know in the chat where you're tuning in from, how many people you're with. We just love to hear more. After the event, I will be sending out an email to everybody who registered with a link so you can watch it after the fact, and also some additional resources that you can explore. So quickly, I just want to give an overview about the Office of Polar Programs. So the Office of Polar Programs is part of the National Science Foundation, which is an independent federal agency focused on funding research and education across core science and engineering. Where we sit within the National Science Foundation, the Office of Polar Programs, or OPP as we call it, supports specifically science in the polar regions. This is anything from astronomy to zoology in these polar regions, and the science can only be done or is best done in these parts of the world. So that would be the Arctic and the Antarctic. So within the Office of Polar Programs, we have the Arctic Science section, which funds research to better understand the Arctic's biological, geophysical, chemical, and socio-cultural processes, and the interactions of ocean, land, atmosphere, biology, and human systems. So our researchers today are aboard the RV Sikuliaq, which is part of the U.S. academic research fleet operated by the University of Alaska Fairbanks. And you can see in this map, and I know Sarah and Beth are going to go into this when they give their presentation, see a little bit about where they are in the world off the coast of Alaska. There's a picture of the ship. So before I hand it over, I just want to give you a quick Intro to our presenters. So we have Sarah Full, who is a professor of geology at the University of Alaska Fairbanks, and she's also the director of GeoForce Alaska. She teaches undergraduate and graduate courses in geology, paleobiology, and her research involves identification of pollen and spores preserved in sediment cores. Next to Sarah, we have Beth Casey, who is a physical scientist at the U.S. Geological Survey, which is part of the Department of the Interior, which is another government organization. And Beth is out of California. She is a paleoceanographer, uses fossil diatoms to reconstruct the extent of sea ice in oceans. And then finally, you will see him in a little bit. We have Josh Barna. He's going to be our tour guide when we get to our ship tour. And Josh is a PhD student at the University of Alaska Fairbanks and is a teaching assistant for several graduate and undergraduate courses. So I am going to stop sharing and I am going to turn over this whole thing to our friends, Sarah and Beth and the rest of the Sikuliaq.
All right. Hi, everybody. We are excited to be with you guys here today because we are indeed in the eye of a storm. Let's back this up a bit and start at the beginning. And we have a few things we'd like to do today, but first is tell you a little bit about why we are here, which is kind of out in the middle of the Bering Sea. Right now, the eye of the storm that we're sitting in is just east of the Pribilofs. We're just east of St. Paul Island. And we've been out in various places in the Bering Sea for about the past 3 weeks. So Beth and I are going to talk a little bit about why we're here. Then we're going to give you guys a ship tour and give you a chance to ask whatever questions you would like to ask about us or the science we're doing or things we have seen or what it's like to be on the ship. So you may have heard of the Bering Land Bridge. It was a lowland region that connected Alaska with Eastern Russia, Siberia during the ice ages when glaciers were growing and sea level was falling. During interglacial stages like the one we're in today, climate is warmer, glaciers are melting back, sea level has risen, and the region that was the Bering Land Bridge is underwater. The southern part is under the Bering Sea. So during the last ice age, some 20,000 years ago, sea level was low enough that Siberia and North America were connected. The shallow part of the Bering Sea was high and dry on land, and plants and animals lived there. And some of them, some animals, some plants crossed between continents, between Eastern Asia and North America, or the other way around. And some of those animals included humans during the last ice age. But the question we are here trying to answer is, what was that crossroad between continents really like? Thanks to fossils in Alaska and Siberia, we have a pretty good idea what those land areas were like during the last ice age. And here's a depiction of what sort of Alaska and the Yukon might have looked like. A wide, cold steppe, a place with virtually no trees or woody plants, covered by herbs and grasses, and it provided happy grazing areas for lots of different animals like mammoths and horses and bison, which are shown here. For a long time, probably the last century, we've often assumed that the Bering Land Bridge was very similar to this, a dry, grassy steppe or a steppe tundra. But in the past few decades, this has been called into question. And the truth is, we're not really entirely sure what the lowlands between Siberia and Alaska were like when they were above sea level. So that is what we're here, out here in the middle of the Bering Sea shelf to find out. I think the name Bering Land Bridge can be a little bit misleading because it makes it sound like this narrow strip of terrain that animals walked or ran across. But the truth is, during the last ice age, some maybe 20,000 years ago, this area is sort of shown in yellow brown here. This is the area that's below sea level today, but that land region was very wide from north to south. It was more than 1,000 kilometers or more than 600 miles. East to west, it could be relatively skinny, or it might also be very wide, depending on where animals happen to encounter it. So yeah, shown on this map also, there's this region, Alaska, the gray part is above sea level today. And of course, this yellow-brown part, the Bering Land Bridge, is below sea level now. Now, animals that crossed this region actually didn't know they were traveling from one continent to another. And in fact, many of them didn't cross in days or even weeks. They lived here. During the last ice age, animals lived here, they fed here. Many of them probably raised babies here. This was their home. And it was their home until the end of the last ice age, when things began to warm up, sea level began to rise, and animals were forced to basically pick a side. Now, because Bering Land Bridge is sometimes a little bit of a confusing term, this region in between what we often call Eastern Beringia, or Alaska and the Yukon, and Western Beringia, otherwise known as Siberia, is often called Central Beringia. And I do tend to use those terms interchangeably. So we're going to show you a little animation of the flooding of the Bering Land Bridge, or Central Beringia, at the end of the last ice age. It's going to start with a very wide, maybe 600-mile north to south land bridge, and we're going to see what happened as glaciers melted and sea level rose. So there's a little, you can see how many years ago are depicted in the lower right, and of course, green is above sea level and blue is rising water. Should start to look increasingly familiar, but you'll notice there also were bodies of water on that land bridge that would have been fresh during the ice age when it was emergent. So there we go, and up to today when things should look a lot more familiar. So one of the big questions we were hoping to answer, and one of the reasons we think that the Central Beringia or the Bering Land Bridge might have been different from Alaska or Siberia during the ice age, is that some animals crossed and others didn't. And some of those animals are named here in these big green arrows with animal names in them. Those are animals that got right up to the edges of the Bering Land Bridge, but did not actually cross between continents. And those include, for example, the woolly rhino, which got up to the edges of Siberia, got into far eastern Siberia, and did not cross. And I think that's a shame, because I think it would have been really cool if there had been a herd of shaggy rhinos living in North America during the last ice age, but it did not happen. The fossils we have on land make that pretty clear. And other animals that were common in Alaska, for example, things like the short-faced bear, got right up to sort of the coast of modern Alaska, may have roamed onto the land bridge, but did not ever cross over to Siberia. And we're really not sure why. It may have been just a matter of timing. Maybe some of these animals got to the land bridge as sea level was rising and encountered rising water instead of a nice pathway between continents or a nice habitat on those lowlands. But maybe also some of those animals got to the land bridge and encountered challenging conditions. Maybe their preferred food source wasn't widely available, or they found terrain that was difficult or impossible to travel through. And the truth is, we really don't know. So why we're here is to explore the Bering Sea in search of sediment and fossils that were deposited on the Bering Land Bridge when it was above sea level. specifically, we're looking for microfossils, and we're going to talk more about what those were, because the way we're going to get the sediment is by collecting sediment cores. And a lot of those cores are about this big. We're both doing it.
Yep.
About this big in diameter or smaller, maybe what, 3 to 6 inches max. So we're looking for very small fossils.
Right. It's unlikely we'll hit something big like
We won't find bones.
a woolly mammoth.
We will not find bones. We are not looking for artifacts or bones of any kind. What we're looking for are tiny remains of plants and algae, which can tell us what the climate was like. And we're looking for them, and specifically, I'm looking for remains of plants that were preserved in freshwater bodies on the Bering Land Bridge when it was above sea level. And on this map, you can see the green stuff is modern coastline or modern land above sea level today. And these bluish blobs out here in the white area that is now the Bering Sea, these blobs are basins on the floor of the Bering Sea. And they're sedimentary basins. They're places where marine sediment is piling up today. And one of the reasons sediment's accumulating there is because these are basins, they're low spots, they're topographic lows. And they've been around for a long time. They've been low spots for a long time. There are some structural controls, movement on faults and things that keep them relatively low areas. And they've been accumulating sediment really for millions of years. So chances are, during the ice ages, over the last couple of million years, as ice came and went, they were low spots on the Bering Land Bridge as well. And water tends to accumulate in low spots. So we are targeting these basins for our coring operations, hoping to go through the marine sediment that has collected in the Bering Sea since the last ice age and get to water that accumulated in freshwater bodies, lakes, ponds, and bogs on the Bering Land Bridge itself. So that's what we're here for. And these are the blobs. We've already spent a little time in collecting cores from the Norton Basin, from St. Matthew and Hall Basins. We've just visited Navarin Basin, and we are right down here about right now heading for St. George Basin. Now, this is some paleo drainage map, sort of a paleogeographic map produced by Jeffrey Bond of the Yukon Geological Survey a few years ago in 2019. And Bond was also making that assumption that low spots, troughs or basins on the seafloor today might have housed either lakes or rivers when the Bering Land Bridge was above sea level, because water seeks low points. And he's got a number of big lakes drawn in on this map. And these are some of the regions we've been exploring. We did look in the vicinity of this low spot in the Norton Basin. We collected some cores sort of east and southeast of St. Lawrence Island down in here. And then we did spend some time out in the Navarin Basin, which is over here. But what we're finding right now is that there absolutely were lakes on the Bering Land Bridge. We have been able to successfully core through the marine sediment that accumulated during the recent interglacial stage and get to stuff deposited in water bodies on the Bering Land Bridge during the last ice age, but the lakes weren't as big as the ones shown here. There were some in the vicinity of these lakes, but what we're really finding is evidence of broad floodplains with maybe meandering or anastomosing rivers, and those floodplains were dotted with ponds and lakes and bogs that are relatively small, or were relatively small. Now, what excites me about being able to obtain sediment from a lake that was sitting on the Bering Land Bridge when it was above sea level is that after this cruise, I can go back to my lab and my students and I can process some of those sediment layers and take out some of the organic matter, put it on a slide and look at pollen grains and spores produced by plants. These things, again, are very tiny. Here's some examples of different pollen grains from different groups of plants, but these things are measured in micrometers, and a micrometer is 1/1000th of a millimeter. So they're very small. And what that means is that like a 1 cubic centimeter sample of sediment can have hundreds or thousands of pollen grains or spores in it. And the pollen grains are products that are distinct to different groups of plants. So you can use them to identify which plants were growing around a lake. And that's awesome, because if I've got sediment deposited in freshwater lakes on the Bering Land Bridge, I can figure out what plants were growing around that lake thousands of years ago. And because plants are very sensitive to changes in climate, I can also look at how those plant communities changed over time and say something about how perhaps the climate was changing over time. Was it warmer or colder? Was it getting wetter or drier? And here I'm just showing an example of a very simple climate index that I created. This was for my research in Mongolia, but Mongolia does have plants that might have been similar to things growing on the Bering Land Bridge. And simply taking something like the percentage of pollen from sage plants, something called Artemisia, the percentage of pollen from goosefoot, which has this crazy pollen grain with little holes all over it, divided by the percentage of grass pollen. These two like it fairly dry. This likes it a bit wetter. So if the number is larger, things were drier. If the number is finer, number is smaller, things were a bit wetter. But you can look at changes in climate over time. And here are some questions that we have been planning to ask, or that we propose to potentially answer as the result of the research we're doing right now and the cores we're collecting right now. These include what plants were present on the Bering Land Bridge during the Ice Age. We're making some progress on that. How did changes in latitude, elevation, and distance from the coast, which was changing all the time really as sea level rose, how did those factors affect the plant communities? And what resources were available to animals, including humans, that lived on the land bridge? And detailed answers are going to take some time, because we need to be able to take the cores that we've got and analyze them for many different proxies, many different things that can tell us about past climate and environmental conditions. But I'm going to give you kind of a sneak peek of some things that we now know that we didn't know before. And this evidence comes from sediment that we've sieved to collect larger bits of organic matter, and from smear slides, which are exactly what they sound like. You take a little bit of sediment, you smear it on a slide, you spread it out in some water, you add a cover slip with some adhesive, and then you can look at smaller things coming out of that sediment. And what we have been finding from some of the layers beneath the marine sediment in our cores, in our sieves, we've found many leaves and stems of mosses. Some of them appear to be aquatic mosses, but they are certainly from freshwater environments. We've also seen occasional birch seeds. And on the smear slides, we have seen moss spores, like this one here, and birch pollen grains. Now, the birch pollen might not necessarily be from birch trees, which might be familiar to you. There is another growth form of birch, which is the shrub. These might well be birch shrubs. There are some differences in the pollen grains, but from the smear slides, I can't yet say whether we had shrub birch or tree birch or both on the land bridge. But one thing I think is fairly clear is that there were woody plants in some locations on the land bridge. And that's something we weren't sure about before. So here's a little graphic to sort of summarize what we've learned so far. This is how we viewed the land bridge for most of the last century. And this is what we think parts of the land bridge looked like based on some of the data we've collected in the last 3 weeks. I'm not saying that all of the land bridge was shrub tundra. I'm not saying that all of the land bridge was boggy, but certainly some of the very low-lying, poorly drained areas were home to plants, woody plants such as shrubs. And there appear to have been some very mossy and boggy areas that maybe intersected the water table and would have made terrible walking for a large, heavy animal trying to cross. Now, clearly, heavy animals like mammoths did cross. There were probably other parts of the land bridge, maybe higher elevation, a bit better drained, that did have steppe tundra or, plenty of grass and herbs for grazing, but it wasn't all the same. And the areas we've been looking at where we see intersecting river channels and lots of small fresh water bodies, lots of small fresh water bodies, we think may have looked something like the Yukon Flats in Alaska today. Meandering or anastomosing rivers on a floodplain dotted with water bodies, surrounded by bogs, with some maybe shrubby vegetation and some herbaceous vegetation. So that's where we're at right now. I'm going to turn it over to Beth, who's going to tell you other things we're interested in out
Thanks,
here.
Sarah. I'm going to turn on this laser pointer. See if
Oh,
that
sweet.
works too. So Sarah mentioned a couple of times these interglacial marine sediments on top of the land bridge. And so that's where my part of this project comes in. I'm an oceanographer. So out here in the ocean, actually looking at ocean sediments is what I do. And the thing that I'm most interested in is sea ice. And so the Bering Sea is a great place to look at sea ice every single year. So this is a map of Alaska and Russia, the Bering Sea here. And this is what it might look like in winter when the sea ice, it gets cold and sea ice comes down from the Arctic and advances across the shelf and freezes over. And then in the summertime, all that ice melts and it melts all the way back, probably off the shelf here up into the central Arctic. And so right now we are out here in the Bering Sea, we're zooming around down here. There is no sea ice out there. We haven't seen a single little tiny ice floe. We haven't seen any snow. We haven't seen any polar bears. If we had instead planned this trip for, January or February or March, it would have been a very different place with lots of sea ice down here. And, maybe we would have seen polar bears by now, but it would have been a lot harder to take sediment cores if we'd done that. So when you think about, okay, so what I want to know is how did sea ice change over time, and how did sea ice change as the Bering Land Bridge was flooding and the ocean was sort of encroaching on that land? And so we're going to look at the ice ecosystem in order to try to answer that question. And so, like I just said, when you think about sea ice, you probably think polar bears, or maybe you like the underdog and you think about seals or walruses, or gray whales, bowheads underneath the ice. But the sea ice ecosystem, none of those animals could even exist if it wasn't for the ice algae that is the base of the ecosystem. So this ice algae, they photosynthesize, they attach underneath the sea ice, and it's super nutritious. Zooplankton will eat the ice algae, and larger fish will eat the zooplankton, mammals will eat the fish, and on up the food chain. And the ice algae also, there's so much of it that it falls to the seafloor, and that's where we get lots of crabs and clams and sea stars, and we're actually bringing up a lot of things called brittle stars in our cores that are similar to sea stars, but they're leggier with less of a body. And so the ice algae is really important in the sea ice ecosystem. This is what it looks like if you were to take a core through sea ice. It's not a lot of sea ice, but just like a meter or so of sea ice. The very bottom of it might be brown. If you were to swim under the sea ice, you might see all of this brown slimy stuff that's like long and stringy, sort of hanging from the sea ice. And if you were to look really close, you would see that the ice algae is actually living in these little channels within the sea ice. And there's zooplankton that are also living in those channels that are eating that algae. And so if you were to look even closer, if you were to melt that sea ice and look at it under a microscope, this is what it would look like. These are called diatoms. They're single-celled algae. And so if you look at this picture, like each one of these little hamburgers here is an individual. Here's another, this is a ribbon of them, and each one of these little stripes is an individual. And so, in this picture, there's, 100,000 diatoms or something like that. Like there's a lot of them. And so they're great for the ecosystem, but the thing that's really amazing about them is that they make their cell walls out of glass. And so because they do that, when they die and they settle to the seafloor, they're preserved really well. And we can actually go back and look at the different species. They last for millions of years in the sediments. So we go back and we look, we're like, okay, what are we looking at? Who was actually living there at the time? We've done some work to try to figure out which of these diatoms live in the Bering Sea when. And so this is a picture of a ship an icebreaker has just gone through here and broken up the sea ice. This is probably early March in the Bering Sea. and the underside of the sea ice is all brown. It's covered in diatoms on the bottom. And so if you look at what those diatoms actually are, this is what they might look like. They look a little bit different from each other, but there's some characteristic shapes that you might see. These guys form these cool little star colonies. But if you were to go out a couple of months later in May, that sea ice has started to melt. There's flows of ice and there's water around those flows. It's a totally different habitat. And if you look closely at those flows, you might actually see a bunch of diatoms sort of living on the surface of the ice or living in the water around the ice. I think this flow is super muddy because I think it had a pile of walruses sitting on it, and walruses are kind of dirty. But if you look at the diatoms here, they look totally different. So here's a ribbon colony. Here's another diatom you might find in melting ice, or maybe somebody that looks like this. And then if you go out in the Bering Sea now, you might find a completely different environment where we have open water, we have no sea ice, and then the diatoms will look completely different again. And so we can really sort of exploit this in order to say, okay, if we look in the sediments and we see species that look like this, then we know that there was open water then. If we look and we see species that look like the ones that were living in the solid sea ice, we know there was a lot of sea ice then, and we can sort of figure out how much sea ice was present in the past. And so here's what we're doing. This is a picture from today. This is what the situation looks like now. And each one of these stars is a place where we've cored. So this is Norton Basin and this is Navarin Basin. And if you sort of roll back time and look at what it looked like 14,000 years ago, these sites are in totally different environments. And so Navarin was probably underwater 14,000 years ago. It had sea ice on it. And so we can look at those marine sediments and look at sea ice. But Norton Basin, it's really far from the coast at this point. It's definitely inland, and we can look at these sediments and think of and be able to figure out what the Bering Land Bridge was like. And the thing that's really cool about that is then we can make connections and we can try to figure out, well, does the amount of sea ice actually impact how wet it was on the continent, or is there no relation? So I have a couple just previews of stuff that we've seen. And so this is one of the first cores that we brought up on deck, and it has a little layer of peat in it, which was super exciting to just be able to see, like with our naked eye, with no microscope at all, that we had hit terrestrial vegetation. This is one of our sediment cores that we opened up. And so it's upside down if anybody out there is actually a purist and knows how to look at sediment cores, but this is the top. These are marine sediments up here, and then all of these stripy sediments are some kind of terrestrial aquatic environment. We don't know. I'm not going to say it was a lake. It could have been a bog. It could have been a pond. We'll have to do some more analyses to really figure that out. But some of the things that we're seeing when we look in the microscope look like this, where this here is a fragment of some kind of plant. And this stack of Oreos here is a colony of diatoms that live, these diatoms like to live right along the coast. And so we can say, okay, this sample is from a very near coastal environment. It's from an environment that probably had fresh water, and maybe it got storms periodically, or maybe it was in the tidal zone and the high tide washed over into it. And this is another sample from also looking in a different kind of microscope that shows a similar thing. So here's a foraminifera. This is a marine organism. It's another type of plankton, but this one is a consumer, it doesn't photosynthesize. And it's surrounded by little bits of moss and plant fragments and some wood. And so again, we're like, okay, here we are, we're in a coastal environment. We have both influences from the marine and from terrestrial environments here. So I think what we're gonna do now, is this the last slide?
Yeah, we're gonna move over to our ship tour.
Yep.
Thanks, Beth. And we had planned initially to do a live tour for our audience here today, but we were worried about losing our internet connection, which does happen sometimes on the ship, and we are pretty far out away from land. So we filmed it in advance, and this did allow us to do a few things which I think have improved the final tour for all of you, because we were able to visit more parts of the ship, and we were able to talk to more people and hear about their expertise. And I think that's really important, hearing from all of these different people you're going to see in this video, because the science on the ship may be driven by the senior scientists' ideas, but in order to accomplish that scientific agenda, we need students, we need the ship's crew, we need engineers, we need technicians, we need people to operate winches, we need technicians, coring technicians who know how to deploy the coring equipment and bring back long cores successfully and have a lot of experience doing that. We need engineers to keep the ship running. We need students to
process our, create fresh water for us and create electricity for us.
Yes.
And actually know how to tell what the weather is that's coming towards us.
Turn this ship into our home away from home for weeks at a time. We need students to take notes and help us actually physically with the core collection, which can be a physically demanding task. We need them to run some of our preliminary analyses. There's a lot of work to be done in the lab as well as out on deck. We need, engineers to keep the ship running and working for us as a living quarters and as a piece of machinery. We need the crew up on the bridge to get us from place to place, but also to hold the ship in one spot while we're coring. And, we also need folks in the galley to cook 3 meals a day for us and keep us fed and happy. So it takes this entire group to help us achieve our scientific goals, and we're really grateful to everyone on this ship for their expertise and assistance.
Awesome. So we're going to start the video, and while the video is going, please feel free. This is a great time to put your questions in the Q&A, and the folks on the Sikuliaq are going to start answering questions while the video plays. But it's going to be great. So here we go.
Hello, I'm Josh. I'm a PhD student from UAF. We're on board the RV Sikuliaq doing some oceanographic research. Next to me is Carmen, a marine tech who works on the ship. Carmen, what are you working on?
This is the CTD. It's the bread and butter of any research ship. That stands for conductivity, temperature, and depth. We measure a number of other parameters, though, like oxygen, photosynthetically available radiation, light transmission. We can also take discrete water samples with these bottles here, and then we can further analyze those samples in the lab. And from this data, we can characterize the entire water column. We can also generate sound velocity profiles, upload those into our echo sounder and multi-beam systems, which just help us gather more precise data.
Interesting. This is really fascinating. Thanks, Carmen.
Sure.
Okay, so now that the CTD is back on board, let's go outside and take a closer look at the deck operations. Safety is very important when you're out on deck on the Sikuliaq. The required equipment is a hard hat to protect your head from things swinging around, a life vest in case you fall overboard, and steel-toed shoes in case you drop something heavy on your foot. It's also recommended that you wear rain gear to protect yourself from any sort of mud that's coming around or water, because you get splashed a lot on deck. Hey Sarah, how are you? This is Sarah, an undergraduate researcher at UAF. An important part of this project is note-taking. Our scribe here, Sarah, has been writing down important details of our project. Sarah, what type of things are you writing down?
So most of the things I write down are the the type of coring equipment we're using, the coordinates, what time we deploy and what time we recover the coring equipment, and also stuff like tension on the winch equipment.
Excellent. That's really interesting stuff. Thanks, Sarah. Keep it up.
Thank you.
Welcome to the back deck of the Sikuliaq. While the CTD deployment was very exciting for us, what we're really interested in is collecting some sediment cores from the bottom of the Bering Sea. If you look over my shoulder, you can see the coring operation in progress. Let's go check it out. Hey, Sam. Sam is a coring technician from Oregon State University. Sam, this thing looks like it should be on the moon. What exactly is going on here?
This is our multi-core. I'm just finishing rigging it up. This contraption is good at getting the top layer of sediment to preserve the interface between the sediment and the seawater. And it goes down and it plugs up the mud, and then when it picks back up on the way back up to the ship, it fires. It fires just like that and picks up the mud.
So in addition to using the multi-core, there are several types of coring equipment we use on the ship. Next to me are Drew and Chris, coring technicians from Oregon State University. Hey guys, what type of coring equipment do you have here?
Well, this we can rig as actually a gravity core or a piston core. So if it's a gravity core, it's basically just a big pipe with a heavy weight on top. So on top, there's 4,000 pounds.
Whoa.
That's as much as like 2 cars or a baby mammoth. And we just lower that into the seafloor. Now at the bottom of it, there's a thing called a core catcher, so the mud can go in, but can't come back out. And then up at the top, there's a valve. So as the seawater needs to go out to let the mud in, that opens up, and then when you pick it up, it shuts, puts its finger on top of a drinking straw. Same thing. Now, if that's not enough to get the mud in the tube, we rig it as a piston core. When we do that, it's gonna free fall into the seafloor the distance of 20 feet or 2 basketball hoops in 1 second. And on the inside of it, there's this piston that's gonna slide all the way up the tube and turn it into like a big mud syringe.
Whoa, that's really interesting, guys. This is really cool. I love this stuff. Thanks. Hey, Chris.
Hey, Josh.
Hey, this is Dr. Chris Mayo from UAF. He's a faculty member at the university. Chris, please tell us about this piece of coring equipment.
All right, well, you learned about the jumbo piston core and Big Bertha. Now this is the vibracore. And the vibracore allows us to collect sediments in areas that there's more sand and small gravels. So it really opens up our toolbox a little bit more and allows us to get the mud and sand that we need. The vibracore works differently. Instead of plowing into the seafloor with 4,000 pounds of weight, the vibracore goes in slow and vibrates the pipe as it's entering the seafloor, allowing us to get cores that are around 6 meters long.
That's really cool. Thanks, Chris. I appreciate it.
Sure, see you, Josh.
See ya. Hey, Sambit.
Hi.
So here we are in the Baltic room on the RV Sikuliaq. It's a staging area between the back deck and the main lab. Here we have Sambit, a postdoc researcher at UAF. Sambit, what are you doing to this core liner?
So this is a core tube where we take our sediment samples, and now I'm leveling it. And so we are trying to make the sections in 150 centimeters interval. And I'm also leveling the stratigraphic top, which actually helps us to identify which is lying on the above and which is lying below of the stratigraphic sequence.
That's really interesting. Thanks, Sambit. I appreciate it. You're doing great work.
Thank you, Josh.
So once we get our cores sliced, we take them over here and put them in the core cutter, and we'll cut the cores in half lengthwise so we can split them open and see what's inside. Here we have Will, an undergraduate student at UAF, and Sarah, a master's student at UAF. They're going to show us how to cut this core in half. So once this core is bisected lengthwise, we can open it up and see what the sediment reveals.
So once we've got it all split up into 150 centimeter sections, we feed each one of those into this pipe and saw it through straight down the middle, going through the pipe, but not the entire layer. We save that for this wonderful saw, which doesn't disturb the stratigraphy as much, so that we can take it into the lab and look at it.
This is going to make a lot of noise, so we have to wear hearing protection and safety glasses, but I'm going to get out of here because I don't really like the noise. This is the wet lab on the RV Sikuliaq. After we finish splitting the cores open in the Baltic room, we bring them in here for some further analysis. This is Sarah, the geoscience faculty at UAF. Sarah, what are you finding in these cores so far?
So after the cores are split, we open them up here, and the first thing we do is sort of clean them off with something simple like this tool, a blank credit card. Clean them off so that we can see the layers. And then we measure the depth in the core and describe all the layers with respect to grain size. Are they sandy, silty, muddy, and color? And by doing that, we can already start to reconstruct how the place where this sediment was deposited has changed over time, because depth in the core correlates with age. The stuff on the bottom is the oldest, and the layers get younger and younger and younger as you go to the top. Right now, we won't know exactly how old those layers were, but we're also looking for bits of organic matter like twigs or other plant fragments that we can send out for radiocarbon dating that will tell us how many hundreds or thousands of years old individual layers in the core are.
Oh, that's really cool, Sarah. Thank you. This core is gorgeous, by the way. I love it.
Lots of interesting layers going and lots of changes over time.
And to my left here is Matt. He's a member of the College of Fisheries and Ocean Science at UAF. Matt, what are you finding over here?
So what I'm doing right now is I'm sieving a small sample from the bottom of this core, and once we've sieved it, we put it under the microscope right here and take a look and see what we're seeing. And what I'm seeing right now is a lot of mosses and small insect parts and other plant pieces that actually don't live in the marine environment. So they're all kind of pointing that it's actually a freshwater or a terrestrial ecosystem, which is really exciting.
This is very exciting, Matt. Thank you. And now, to my left here is Ryan, a PhD student at UAF. He's taking things a little, looking at things on a little smaller scale. Ryan, what are you up to?
I'm looking at smear slides, where we take a small amount of sediment from those cores and smear it across a microscope slide, where we then will take and look it under high magnification, and we're identifying diatoms and pollen to see whether or not the depositional environment was freshwater or marine.
This is really cool stuff, Ryan. Keep it up. I love it. Hey, Beth. So this is the main lab on the Sikuliaq. Next to me is Beth Casey from the USGS. She is working with the multi-sensor core logger. Beth, what's going on with this thing?
So before we cut the cores, we take the cores in, we line them all up here on the table, dry them off, and we feed them through the scanner. And the scanner, we've got a couple different sensors here. This one actually shoots a little bit of radiation through the core to figure out the density of the core. And then this one determines whether there's any magnetic minerals in there. And all this data comes up on the computer screen right as we're scanning it. And so we have access to that data right away to see what's happening in the cores and how they look different or similar to each other.
This is so interesting. Thanks, man. The main lab on the Sikuliaq serves multiple purposes for the science team. We store all the cores that need to be scanned on this table. They're staged here. This is where we have our meetings, and we basically lounge here when there's some downtime. We also have a ping pong table for our entertainment. Chris and Jenny, how are you guys doing?
Good. We're having a good game here.
Looks nice. Jenny, come on, you got this. So
It's hard when the ship moves.
it is fun. It is very hard when the ship moves. I am undefeated so far on this cruise, so I'll take all comers. On the wall here is our board of lives. This is where we have all our information for the day. You can access it via this camera in your room if you don't want to get up early. And the most important part of scientific research is good bookkeeping. Here we have a bank of computers for people to enter their data for the day after logging cores. And we've got some people working on some research where to go in the future for future coring sites. We are now in the computer lab on the Sikuliaq, where a lot of the geophysical stuff takes place. Next to me is Jenna from the USGS. Jenna, how are you?
Good.
What are you up to?
So here in the computer lab, we have a lot of different sensors on this ship that help us figure out the shape of the bottom and what's beneath the seafloor. So we use different things like multi-beam sonars, which allow us to map the depth of the ocean and figure out the shape. And then we also have different types of sonars and that use sound to map the layers of sediment beneath the seafloor. And with those, we can look for different features that we actually want to collect sediment samples from. And that's what Sarah's looking at on this screen over here. She's going to show us some examples of what this looks like and what the kinds of features that we're looking for.
One of our duties as scientists here on the ship is to keep watch of these screens for 24 hours a day, and we do it in shifts of 4 hours. And we basically look at this data here and we look for interesting features. Right now it's really boring, but what we're looking for is stuff like this. This is a little basin that we cored just today, and it has this like nice little U shape here, and we're looking for features like that.
Thanks, Sarah. That's great. I appreciate it. And Jim, what are you doing over there? I see you're sitting at a computer as well.
In addition to the sub-bottom profile that Sarah spoke to, we're also looking at a litany of other screens here, giving us a bunch of different information from chemical and the underway data display to some weather. In front of me is the OLAX, which gives us kind of our heading, our current position. And of course, above that, we have some of our multi-beam data, chirp, all the pretty colors up top. And up and to my right is a few different camera views that we have, along with some winch data just above the OLAX here.
This is Tony. He works for OSU as a technician. So how many winches are on board this ship?
So there's these two in this room, and then there are hydraulic power packs, and then we have another really fancy winch behind us on the other side of that wall, a couple bulkheads away, and that's our traction winch.
Or how many meters of cable do you usually carry on the ship?
The traction winch standard layup there is 10,000 meters, so we can get to like full ocean depth, 5,000 meters, 4,000 meters, and then we still have enough left over to tow things.
I love this room. This really speaks to me. Thank you, Tony. I appreciate it.
Yeah, you bet.
The Sikuliaq also has a treadmill for those who want to stay in shape. Currently, the seas are pretty high, so it's dangerous to run on this. And so I'll just keep standing here. Also downstairs is a complete gym for those who want to work out. And upstairs, we have a sauna next to the lounge. So the science crew on the Sikuliaq works 24 hours a day. We have two 12-hour shifts, so we have to be extra quiet while we're in the hall here. Let's take a look inside of one of the staterooms.
Hello, I'm Sochi, and this is the stateroom I share with Sam. This is my bed, and that's Sam's bed. And here we have a desk, and it has one of those nice anti-slip mats that the mess hall does. We also have a lot of cabinets to lock our stuff away because things will obviously roll off on a ship, and a TV where we can switch between different cameras on the ship to know what's happening from the room. We also have our sink here, and the toilet and shower are inside this room, which we share with the room next to us. We have to remember to lock both doors when coming in and unlock both doors when going out.
So this is the lounge on the Sikuliaq. We have a bunch of recliners, lazy boys for people who want to come in and relax after a long day at work. We have a large screen TV. We have a library full of books, and we have cases and cases of DVDs for us to watch for our entertainment and on our downtime. Here we get all of our meals served to us 3 times a day. We have breakfast, lunch, and dinner, and there's a fridge for snacks. Today we are having halibut chowder. All of our meals are prepared in-house. Everything is delicious. We have fresh fruit and vegetables, the whole cruise, and I've not gone hungry yet, and I think I'm gaining weight. There is a secret freezer that is full of ice cream. It's not really a secret because the ice cream supplies are dwindling rapidly. I wish it was a secret because there are some Big Bopper ice cream sandwiches that are gone. Hey, Christoph.
Hello.
We're up on the bridge deck of the RV Sikuliaq. This is the chief mate, Christoph. Christoph, how are you?
I'm well. How are you doing?
Oh, great. Thank you. So, a few questions for you. We're up pretty high. How high are we?
We are very high. We're up 50 feet above the water, and we're 4 levels above the science labs.
Interesting. Okay, yeah, that was quite the hike up the stairway. So, what else do you see from up here?
We get to see all kinds of things out these big windows. We're looking for ships and boats, other things that we might have to maneuver around. We get to see all sorts of animals up here. We get to see whales, dolphins, seals. When we're further high up north, we can see walruses and polar bears.
I love the view up here. So, interesting question, or weird question. I don't see one of those big old-timey steering wheels. How do you drive the boat, or the ship? Sorry.
Right, we don't have a wheel because we don't have a rudder. Other ships will use a rudder to direct the water flow from the propellers. Instead, we have azimuth thruster pods, which can spin all the way around 360 degrees, and we control those using our what we call combinators right here.
All right, Christoph, one last question for you. Can we please hear the whistle for the ship?
Absolutely.
I love it. Thank you. That's great. Okay, so that concludes our tour of the RV Sikuliaq. I want to say thank you to the crew for allowing us unlimited access to the ship, and also to the science party for their good spirits and letting me talk to them while they were busy today. And I want to say thank you finally to JR and Natalie for their great work filming us today. This has been really fun. Now let's go back to the science crew for some Q&A.
Awesome. Oh gosh, now we are nice and close with the rest of the Sikuliaq. Thank you guys so much. I loved that tour. I also was imagining a giant wheel steering the boat. So I know we're running low on time, so I want to get some rapid fire questions as much as we can. We might go a little bit past the top of the hour. And so for those of you who have joined us, please feel free to stay as long as you can. I do want to also shout out the social media account for the ship and for this project. You should be able to get tons of answers from that as well. So let's just start off with, are you able to get DNA from the pollen or spores or different samples that you collect from these cores and recreate genomes or anything else from ancient plants?
Yeah, this is Matt here, and that's a great question. We're actually collecting special cores that are going to be specifically used to analyze ancient DNA from the cores. So, yep, we hope to do that.
Awesome. Now, we got a lot of questions from a classroom in Nevada, and so shout out to them. I'm sure they're nice and warm. And one of the questions they have is about carbon bubbles and climate change. And so I'm wondering if you could address, one, do you see like bubbles of air in what you're collecting? And then also, how has climate change affected the work that you guys are doing in this region? Since I know some of you have been around doing many years of cruises.
I think one thing we can say about climate change affecting our work is that even a few years ago, we would have had a fairly storm-free month in August out here on the Bering Sea, and that is not the case this month. The storms are coming earlier, partly because things are warmer and there's sort of more energy in the system. But
I think the other thing, I feel like climate change motivates the work. So we want to look at a period of time when the Earth was warming in the past. And so when you think about, we're going from a glacial into an interglacial in the past, this is a natural warming. And it's a good experiment to sort of look at how do things change as that warming happens? How do things change on land? How do things change at sea? Obviously, it's not exactly the same because all the ecosystems are slightly different, but it's a good little experiment.
yeah, it's a good question, because one question we have is how quickly did sea level rise during this period of natural warming, providing a sort of a basis for comparison to sea level changes we're seeing today.
Yeah. And also, we're not seeing gas bubbles in the cores, but we do have some gassy cores. And so there's gas that we can see on the sonar that sort of obscures what the sediments look like below. And then sometimes when we take those cores up, they're a little stinky. And sometimes the gas tries to escape. And so it pushes the cores apart a little bit and they may fall on the floor or something.
Mud tries to get out of the tubes as we cut them up. It's pretty interesting, but probably that's from decaying organic matter deeper in the sediment.
Yeah.
So kind of related to that, Lucy, age 10, asked about getting cores that are unusable. So if you were to get one of these cores that you're just describing, what do you do at that point? Or do you get cores that just don't work?
Yes. I learned this early on because most of my coring experiences was much smaller equipment. And then you do your best. Sometimes there's still things you can get out of cores that might have somewhat disrupted stratigraphy, or the layers might have gotten a little mixed up, or the core might have gotten pulled apart by suction on the piston as it was coming up. But you still cut them up, cap them, and keep them for any information they might contain.
Awesome. So when you were showing your maps, you showed this like giant continent essentially, and I believe it's Beringia. Is that how it's said? This whole area?
Beringia.
Yeah. All right. Fantastic. Do you have a sense of maybe when scientists will be able to talk more regionally within that kind of historic ancient continent, or are we not there yet?
Rather than generalizing to the whole continent of Beringia. Yeah.
That's what we're trying to stop doing. And we will be able to talk about specific regions of the land bridge itself, or central Beringia, I think, following this cruise, because we're certainly finding that while the land bridge may have been different from eastern or western Beringia, often it mattered what the elevation of the land bridge was, what the gradient or the slope was toward the sea, how far from the coast various sites were, and at different times. So we hope to be starting to get into a more regional view instead of assuming the whole area or even the whole land bridge was the same.
All right, so I'm gonna a few more questions right as we get to the top of the hour. First of all, I want to let you know that one of our attendees says that you all deserve ice cream. So please go enjoy. I think it's
Thank you.
I think it's noon time there, so lunchtime, so you can have ice cream. Even if it were at 4 a.m., you should have ice cream. How many people are there on the ship, and like kind of how big is the ship in terms of maybe feet, or I don't know if we have a ship tech up there with you guys. Or a general estimate. Oh, we have someone all the way in the back. This is great.
Hi, yeah. So I'm Jenny. I'm a marine research technician for the Sikuliaq. And so we have 48 berths on board. So that's 22 crew. Then there's 2 science technicians, and then there's 24 science berths. We don't quite have a full cruise. So I think we're around 45 people for this trip. But we have the capacity to do 48.
And when you say berth, you mean bed?
Berth meaning a bed.
Yes.
Yeah.
All right. For those who aren't as familiar with ship terminology, outside of the head, right?
Oh yeah. How long is the ship? Oh, the ship. I always mix up this number a little bit. I think it's 276 feet long. 261.
All right. So fantastic. So I'm going to close this out with this question that I think is really interesting, just about how long it takes to do your science. I mean, I think we watched that video and it was like, boom, 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 core it, cut it, look at it under the microscope, and then you're done, right? I know that's not the case. So how long on average would you say it takes to process kind of a core sample? You know, are we talking, I assume, months and maybe even years?
Well, we're heading to Oregon State University in early October to do most of the processing. And it's going to sampling, excuse me. So it's going to be taking months to years, especially going through the fine scale microscope work. It's going to take years.
Yeah, Ryan is just starting their PhD, so maybe is being a little optimistic. This project has been sort of in the brains of Sarah and me and Dave Schull and a few other people, Jenna, for a really long time, for upwards of almost 20 years. And I expect that, you know, at minimum, we're going to have 3 or 4 years of intense working on it. And then these cores will just keep on giving for years after.
Yeah, I was just gonna say, I think one of the things that's really exciting about a cruise like this is that we make discoveries when we're at sea. We learn new things when we're here, and then we go back and we work on the course for months to years. We learn new things then, and then they get in archives for years. And then other researchers come along and get to use these samples and get to ask different kinds of questions and build on the science that we're doing. And so it's something that lasts for decades, you know, and hopefully even centuries, you know, that we start to build and build and build on the science. And that's one thing that's really neat about it.
But I did see that we had some questions even in advance of this broadcast about dating, and that is going to be one of our top priorities. And when we are at OSU in early October sampling, we're going to be pulling out bits of organic matter, preferably terrestrial organic matter, but in the marine section, we might do something like forams. Yeah, I know. I'm thinking only about the Bering Land Bridge itself. We'll pull out plant bits, and we'll pull out marine organic matter, and we can send them off for radiocarbon dating. And we hope by the end of this year, we'll have a better idea how old different horizons in the cores are.
Yeah. All right, and to end it with just a really quick one, Jenna, what are you holding?
This is Dr. Narwhal. Dr. Narwhal has been sailing with me at sea for several years now, and she's an important part of the science team.
Well, we're happy to have Dr. Narwhal on board. I don't know if Dr. Narwhal gets her own birthing unit or not. So I do want to thank all of you for letting us in on what your life is like on the ship, the science going on. Thank you for showing us what your rooms look like and the kitchens. And I do want to just end by sharing a brief slide with everybody who is still here. You can follow all of our accounts here, the Sikuliaq, the Bering Land Bridge Project, and the National Science Foundation on Twitter, Instagram, Facebook. You can also visit nsf.gov. After this event, I'll be sending an email that has tons of links, many more than what we have here. And you can ask questions and engage with all of our scientists and all of the really cool things going on in the Arctic. And so thank you all so much for joining us today. We hope you have a great rest of your evening, morning, whatever time zone you are in. And to the folks on the Sikuliaq, I wish you smooth sailing.
Yeah.
Sharing. We're just gonna wait 1 moment. Check something. Right, I think there was an issue with YouTube, so if there was, then we can wait for all of our attendees to go and then